Alright, so we have just finished up cell division and binary fission. You guys should have taken your quiz by now. You should have also watched your video and responded to the Intro to Genetics assignment. So you should know some of your key terms. You should have done your crossword. So this stuff that I'm about to go through should be pretty familiar. We're going to go through the key terms from the crossword and the notes and the stuff that you've been doing already, and then we're going to move into learning a little bit more about Punnett squares so you guys can get some practice with Punnett squares. So an allele is different forms of a gene. We know that a gene is a section of DNA on a chromosome that codes for a particular trait, and since we get for each trait, we're going to get it from our mom and from our dad. Okay, that means we might get one form of an allele from our mom that's different from that form of the allele from our dad. Okay, so for example, if the gene that we're looking at codes for whether our earlobes are attached or detached, like you see in this picture here, then maybe you have attached earlobes or maybe you have detached earlobes. So maybe go ahead and check, look at this picture and see which one of these you think you have. You can also look at your parents and see if you look like them. So attached earlobes are a recessive trait. Okay, so we're going to use E as our representation for these alleles. So since attached is a recessive allele, it's going to be represented by a little e. And then since detached are dominant, we're going to represent that by a big E. So if you have attached earlobes, and that must mean you got the attached earlobe from mom and the attached earlobe from dad, and that's why you have attached earlobes. If you had gotten the detached earlobe from dad and attached from mom, then you would still have detached earlobes because detached earlobes are the dominant trait. And then obviously if you got detached from both parents, then you would end up with detached then as well. Okay, so if you take a look at your parents' ears, theirs might, even if you have attached earlobes, maybe they both have detached. That just means that they each carry a recessive trait. That means that both of your parents must look like this, and that you inherited this from both of your parents. Okay? So that is what an allele is. It's different forms of the same trait. So the trait would be what your earlobes look like, and then the different alleles would be whether they're detached or attached. Different coding will code for those different alleles. Okay? Locus, that's another new word for us. So if say we have this first pair, okay, so remember we have 23 pairs of chromosomes. This one's going to be from mom, this is going to be from dad for our first pair. Okay, so the locus is a specific location of an allele on the chromosome. Okay, so we'll say that this section is the gene and it matches up with this section. So it's that location where the gene is. That's what a locus is. Okay, we have dominant versus recessive, which we just talked about. Dominant alleles are going to be expressed. You're going to actually see them. Okay, you only actually see a recessive trait in someone's appearance if they received that recessive trait from both of their parents. That's the only time that they would actually express that recessive trait. Okay? Alright, so now we have heterozygous versus homozygous. So let's bring this down here. For heterozygous, let's break down this word. Okay, so hetero just means different. Okay, and what this means is that the alleles that you receive are going to be different from each other, which means that you're going to receive one dominant and one recessive. Okay, so if that organism has one dominant allele for that trait and one recessive allele from that trait, then we would call that heterozygous. They got a dominant one and they got a recessive one. So the two alleles that they have for that trait are different. Okay, then we have homozygous, and homo means same. So organisms that are homozygous are going to have the same allele from both parents. So either they got a recessive allele from both parents and they are homozygous recessive, or they got a dominant allele from both parents in which case they are homozygous dominant, and they will express the dominant trait. Okay? Then we have the terms phenotype and genotype. Genotype are these letters that we're looking at. They are the actual DNA and how we would express that DNA. So the genotype are the genes that people have. The phenotype are the actual physical characteristics. You can remember that because physical starts with a PH and phenotype starts with a PH. So the person's physical characteristics, which are controlled by their genes, are going to be termed phenotype. Okay. Then we have codominance. This is a little bit tougher, okay, because it's easy to understand dominant and recessive. But when it comes to codominance, it's exactly what it sounds like. It's where you have two alleles for a particular trait that can both be dominant. Okay, this is best understood through learning about blood typing, which we'll learn about more when we get back to school. But with blood typing, you have probably heard that some people have A-type blood, some people have B-type blood, some people have O, and then you've probably also heard of AB. Right? So this actually, blood type is an example of a couple of different terms. It's an example of codominance because AB blood is when the organism receives, or the human receives A blood from mom and dad, or dad, and then B blood from mom or dad. And instead of one masking the other, they actually just mix together and are both dominant. Okay. Another thing that's expressed here that we haven't learned about yet is the fact that there are multiple alleles. So there's not just two options. A and B are not the only two options. You also have O, which we'll learn about later. But this is codominance. AB blood typing, which is where A and B are both dominant. That's why we write them both in the expression there. They are both dominant. They're both going to just kind of be mixed together instead of one being masked. Then we have carrier, which is another term we've learned about. Someone who's a carrier, they have an allele that is not actually expressed in their phenotype. So, for example, maybe you have attached earlobes, which are recessive. So your genes, your genotype would look like this. Well, maybe both of your parents have attached earlobes, which means that they must each look like this when we look at their genotype. So they were each carrying an allele that was recessive. So even though they have detached earlobes, because the dominant trait is what's expressed, they were still carrying the recessive one. So they were able to pass it off to you, even though in their phenotype, the dominant is what's expressed. You received both of the recessive. Okay, so they are carrying that trait even though it's not expressed in their phenotype. Okay, so now we're into Punnett squares. So first what I want to talk about is just kind of some basics of Punnett squares. We will get more in depth into this when we get back into class. So there were some Punnett squares in the video that you watched. Okay, let's stick with earlobes just because that's the example that I've been using. You saw a different example on your video. Okay, so we said that little e is obviously going to be the recessive trait, and that's attached. And then big E is going to be detached, and that's going to be the dominant trait. Okay, and we will say that the mom here has big E, big E. So she's homozygous dominant for detached earlobes. And then the father is homozygous recessive for attached earlobes. So what we do is we're going to match these guys up. So since this E is located right next to this row, we're going to fill that little E in across. And the recessive trait always goes last, so we'll fill it in like that. Okay, we're going to do the same thing with this other little E. And then we're going to take this big E down with the column that it's aligned with. It just so happens that because each parent was homozygous, we're going to end up with a totally uniform potential genotype for the offspring. So what we're looking at here is all of the different combinations that the offspring could get. So if these two organisms were to go through fertilization, then you would definitely end up with a heterozygous genotype, no matter what, okay? Because these are the only alleles that this person has the potential of passing on, and these are the only alleles that this person has the potential of passing on. So no matter what, they're going to end up with an offspring that is heterozygous. And since they're heterozygous, the dominant allele is going to mask the recessive allele, so they are going to have detached earlobes, and they're going to be a carrier for the recessive trait, which is attached earlobes. So they could still pass it off to their future offspring. Okay, let's go ahead and try another genotype here. Okay, so let's say that the parents are, one parent, the dad, is heterozygous, and the mom is homozygous dominant. Okay, so we're going to do the cross just like we did before, filling in the rows and columns. Same thing here. Okay, and what we come out with is, for our genotypes, okay, we have 50% are going to be homozygous dominant, and 50% are going to be heterozygous. Okay, but when we talk about the phenotype, since they all have that dominant allele, they're all going to have the dominant phenotype. So they are actually all going to end up with detached earlobes. Okay, because they all have that dominant allele. Okay, so if these organisms go through sexual reproduction, then they are going to end up with an offspring that has one of these 4 genotypes for this allele. I'm sorry, for this trait. Okay, all right, let's try another one. Okay, so for this next one, we'll say that we have heterozygous here for the dad, and then homozygous recessive for the mom. Okay, so we're going to fill this in, fill in the rows and columns just like we did on the last one. Okay, and this is what we're going to end up with. We end up with, okay, we're going to end up with the genotypes being 50% big E little e and 50% little e. So these organisms, as far as the phenotype are concerned, are going to be 50% of the dominant allele shown, the detached earlobes, and then 50% are going to have attached earlobes. Alright, so that's what can be passed off in those options. And we could have different traits, different options here. Okay, so there's multiple different options that you could have. It all depends on what the parents have as to what they could potentially pass off to their offspring. Okay, so you're about to do some practice with this. I have some problems that I've posted to Moodle, and you're going to go ahead and give those a try. Those are going to be due by before midnight, 11:59 on Friday night. Hopefully we're back in school and we'll have time to work on them together. But these are going to be due as soon as you can get started on those would be good. All right. Email me to set up a Skype conversation if you have any questions.